Hi, everyone. Today, we are going to continue with layers of the retina. We are looking at the anatomy of the human eye. We're looking at special senses. All right. So now, allow me to go through layers of the retina. So what you are seeing on the right side of the page or the slide, this just shows the different areas of the retina. Okay. So these are which have been demarcated in certain parts. Yeah. So we have what we call photoreceptors, then we have both pigment epithelial layers, and then you have both the bronchus membrane and the cochlear capillaries, as you can see. But we'll go in detail as you go on. So allow me to go through the text. So cells in the retina are epithelial, photoreceptors, neural, and ganglion cells. Okay, so these are the different types of cells that are found in the retina. Okay, so basically we're looking at how can vision be achieved. Okay, so we're looking at neurophysiology, to be more precise, spatial senses, the anatomy, the human eye. So they are arranged to form the retina layers, these areas that I've mentioned in bullet number 1. I mean, those parts that I've mentioned in bullet number 1. Okay, so now the layers start with epithelial cells in the outer layer that are set on a myeloid pigmented layer. Okay, so when you talk about the myeloid, so the myeloid has to do with, as you know, we had the choroid layer. So the choroid layer has got what we call melanin. If you remember in the first video, I did talk about how this pigment is capable of achieving the function of absorbing light, okay, or UV light, which can be harmful to the eye or to the body. So now, the epithelial cells have tight junctions between them and host. The epithelial cells have tight junctions between them and host the other outer segments of the rods and the cones. So remember again, we talked about the rods and the cone cells. We said these are light sensitive cells. So we'll talk about them more in detail as we go on. All right. So now the photoreceptors synapse on what we call the bipolar cells, with the multiple rods synapsing on a single bipolar cell, while a one-on-one is noted in the cones to bipolar cell. So we talked about, I think in the previous video, we talked about how these synapses are. And I believe by now you know what a synapse is, just a junction between two adjacent cells, which create a communication junction, okay? So now, when you look at these are the different types of cells that you're talking about. So as you can see, we have got what we call the cones, all right? Here, the ones which are being depicted in blue. And we have got this other type, which is shown, which are known as the rod cells, okay? So blue in this slide, it's being depicted by, purple is the one that's showing cone cells, while the rod cells are this one. So even if you can see these types of cells, they have got different structures, and these different structures are going to be very important as we go on. So it's one of the distinctive things that you can be able to see, and in terms of how they function, when we talk about the physiology that is involved in achieving a clear image, or in achieving what we call sensation of light, okay? So now, so we talked about the light sensitive cells, which are known as the rods and the cone cells. Then after that, we are going to have what we call the bipolar cells. So if you can see, the bipolar cells are forming a synapse with what we call the light sensitive cells, the rods and the cones, as you can see, all right? These are the bipolar cells, right, that you're able to see. Then after that, we are going to have what we call the ganglion cells. We are going to have what we call the ganglion cells. And these ganglion cells are forming a synapse. The bipolar cells and the ganglion cell is a synapse. All different types of cells which are found in a horizontal position. So you can see that we have got what we call the horizontal cell there, and also have got what we that are found, or that are also synapsing with these bipolar cells and ganglion cells. These are known as the macrine cells that you are seeing here. Then there's also another cell known as the horizontal cell that's being found at the synapse between the cones and the bipolar cells. So please pay attention and follow step by step, because this is going to be very important in trying to interpret how light is transmitted, or how it's going to be interpreted in the right pattern, right way. Then when you see here, there's light coming in, okay? So light comes through this direction, then it goes all the way up to the light sensitive cells, then it will start moving in this, starting from the cones or rod cells, then bipolar cells, and after you're going to have the ganglion cells, which also synapse or join, okay, to form what we call the optic nerves, okay? Yeah, which transport the signals to the brain for interpretation. So that's what this is saying. So like the text was saying, the photoreceptor synapses on bipolar cells with multiple rods synapsing on a single bipolar cell, while a one-on-one is noted in the cones to bipolar synapses. Both the bipolar cells and the photoreceptors produce graded potential. I'm sure by now you know what a graded potential is, so there's no need for me to go through it. So now the BP, the bipolar cells synapse on the ganglion cells, which can produce an action potential. AP means action potential, and whose axons go out through the optic disc. Okay, so optic disc, we said that's where the optic nerves are leaving the eye, and no light interpretation can happen there because you don't have either cones or rod cells. You only have optic nerves. Then there's a neurotransmitter that is in the conducting cells, is known as glutamate. So glutamate, we are going to talk about it as we go. It's very, very important, and it's very, very cardinal to allow light perception or interpretation of the information that is coming in form of light energy. So remember, what you're talking about, the chemical, the neurotransmitter that's going to be found on this synapse is known as glutamate. Okay, now, local connections are noted within the retina, include non-conduction cells that have other effects. All right, so local connections are located within the retina, include non-conduction cells that have other effects. So the horizontal cells connect the bipolar cells, and areas in which they connect make a plexus or outer plexiform layer. In the inner layer, the junction of the bipolar cells and the ganglion cells are horizontal, connected by amacrine cells, making the inner plexiform layer. Okay, so in the previous slide, I did talk about this, so there's no need for me to go in detail. So cells connecting the two plexi are noted interplexiform cells. So the horizontal cells are the GABAgenic, and so are amacrines, which can also be glycogenic or chologenic or dopamagenic. So they're just talking about what type of neurotransmitter they can be able to produce. So now, when you look at this, we have got what we call the rod cells and the cone cells, okay? So in between, we had horizontal cells, which are able to form a synapse in between, okay? Yeah, and here, all right, as you can see, this is a horizontal cell, it's able to form synapses there. And there's also another one, which is known as the amacrine cells, which are able to see here in yellow, okay? Yeah. All right. So ganglion cells in the retina keep the layers intact and also insulated, okay? So the ganglion cell in the retina keeps the layers intact and also insulated. So these include the Mueller cells, which keep the rods and the cones in place with their cilia forming a layer. Outer limiting membrane. So there are processes that go to the top of the layer forming food plates, inner limiting laminar stroke membrane. So there's also what we call astrocytes, are star-like, or the star-like and the supporter cells. So if you go back when you learned about in anatomy, probably in an introduction to cell physiology, about it before you started learning about the tissues, I'm sure you did talk about what astrocytes are and what they do. We also talked about microganglia and microphagus and spread through the layers, okay? So now, this is also another image trying just to depict what we are talking about. So now when you look at this, you're going to realize that we have the retinal layers there, right? And other components. So we can start from here, okay? So we have the pigment epithelium, as you can see. Then down there, we have what we call the choroid. So basically, you're still on anatomy of the eye before we start the physiology. The physiology is very nice once you appreciate the anatomy, because we'll be talking about how a nerval impulse or how information flows, starting from the light-sensitive cells, going to the bipolar cells, again to the ganglionic cells, until they are transmitted via the optic nerve. So no wonder that's the reason why we are learning about this. So we have got the choroid, which is there. We said it's a melanoid tissue, meaning it contains melanin. Then after that, there's what we call pigmented epithelium or pigment cells. Then these are the different types of cells which are light-sensitive cells. So as you can see, this is known as the cone cell, these are known as the rod. So two photoreceptor layers. So there's this one, okay? Then after that, you're going to realize that these are also photoreceptors. 3 outer limiting membrane, okay? Then there's 4 outer nuclear layer. Then there's 5 outer plexiform layer. Okay, you remember outer plexiform layer where we're going to find the horizontal types of cells, the amacrine cells. Then after that, you're going to go into what we call the inner nuclear layer. Then 7, the inner plexiform layer. Okay, so then 8 is going to consist of what we call the ganglion cell layer. Then we have got 9, the nerve fiber layer. Then 10 at the end is known as the inner limiting membrane. So now when you go into this, you're going to find that these are pigment cells, cones, rods, as the arrows are depicting. There's a horizontal cell, amacrine cell, bipolar cells, and all these things that you are seeing here, we have already talked about, and it's just an improved image that you're able to see there. So now cones and the rods, okay? So we'll talk about what their anatomical features are, and later on, we'll appreciate their physiology. Now, cones and rods, these are the receptors of light. They receive light as the stimulus that allows them to conform and transmit impulses to the brain centers, okay? So they are sensitive to light, okay? So like we discussed later, in the previous video, that the rods are responsive to dim light while the cones are responsive to bright light. Hence, this means that you need a lot of work to stimulate the rod cells, okay? While, you need a lot of work to stimulate the cone cells, while the rods only require about 1 photon of light for them to be stimulated, okay? So now, they receive light as a stimulus that allow them to conform or transmit impulses to the brain centers. So cones are responsible for daylight vision, which makes sense because they need a lot of light for them to be stimulated. So they have got higher acuity, mediate color vision, and more on the fovea centralis. That's where they are found, which is the opposite of the rods. So rods are highly sensitive and can detect dim light. So the highly sensitive, what do you mean? Only 1 photon of light can be able to stimulate them. While else the cones, they take more stimulation. Like that's the reason why they are responsive in bright light or during the day, because they need a lot of light, while the rods only require dim light for them to be stimulated, or they're very sensitive. Sometimes I give this example of ladies, and there are women that we can call their easygoers, whereby a man is not supposed to stress a lot in order for him to win their heart. Yeah. So those can be like rods. They just require someone to just show interest and they already fall in love with that person. While the cones, they need you to put extra effort to show that you're interested in them. So that's the examples or lame examples that you can give in order for you to remember. So rods are highly sensitive and can detect dim light. So loss of rod results in night blindness and loss of peripheral vision, okay? So loss of rods results in night blindness and loss of peripheral vision, meaning it's difficult for you to see at night because they are most effective at night. They need only fewer photons, like 1 photon of light can stimulate a rod cell, okay? And peripheral vision means side vision. They contain more photosensitive pigment than the cones, so more in the peripheral area. So we said the cones are more concentrated on what we call the fovea centralis, while hence the rods are found scattered around the retina. So what you are able to see, the rod photoreceptors, and these are the cone photoreceptors. So even if you look at the shape, if you can see, these are different, and this will be very important as we go on. So they have got about 3 segments. So I've got the outer segment, the inner segment, and the synaptic terminals. So now when you look at the outer segment, the rods and the cone cells are different. Okay, now other differences of the receptors. The rods are mostly on the periphery while the cones are central. At low illumination, okay, meaning low light intensity. So at low illumination, the rods are more stimulated as they have a low threshold. Easygoers, okay? Yeah, it doesn't take time. So now in rods, even lower light can activate them, and the graded potentials can submit to activate the bipolar cells. Remember, we have to start with the rod cells or light sensitive cells, then bipolar cells. Then after bipolar cells, you have what we call the ganglion cell, then the ganglion cell, then we have the optic nerves. So the more sensitive receptors are therefore the rods. The response to difference in wavelength is limited. So since cones are mostly one-on-one synapse, they do not easily activate bipolar cells. They are specific to what illumination they are conducting when activated. So cones that have 3 types that respond to 3 different wavelengths. So that we're about 3 different wavelengths that can the cones are able to respond to. Then that with a higher frequency of vision is noted when there is a higher number of cones. Okay, now structures of receptors. So the structures of receptors, as you can see, that's what we're talking about over and over again. So now the outer segments are modified cilia. Okay, so now when you look at the shape of the outer segment on both cones and rods. Okay, so this is looking like a comb. Okay, yeah, for ladies, I think they have seen a comb which looks like this. So this is an example of the cones. Wow, if you look at these, the structures on the rods, they are closed up. Okay, in spaces in between. So that's how they look. So outer segments are modified cilia. So outer segment, there's inner segment and synaptic terminal. Please pay attention. So now, made up of regular stacks of flattened circulars or discs. They are renewed in rods constantly from in to out, while in cones, their multiple sites are renewed. So contain photosensitive compounds that react to light. So talk about the physiology. That's where the physiology comes in. We talk about compounds that react to light. What makes it? Remember, when we talk about sodium and sodium will come again. No wonder I always encourage my students to pay much attention in cell physiology, because once you understand cell physiology, that means you have understood the backbone of physiology. Okay, so they initiate action potentials in visual pathways. Okay, they initiate action potential in the visual pathways. We talk about the pathways, the visual pathways, or the mechanisms which result in producing an image which is clear and which can be interpreted properly. Okay, so these are the rods, as you can see. Okay, then these are the rods and the cones. Then this plasma membrane, as you can see there. In the outer segment, there are discs or sacs. Then the serine neck, as you can see here, serine neck. Then there's mitochondria and the nucleus. So whenever you say mitochondria, that means it's going to be some active transport somewhere, because energy is needed, or ATP is going to be needed at one point. Okay. Now let's go to the visual process. So visual process is a series of actions during visual perception. Okay. So this just talks about the mechanism on how we can be able to perceive or be able to visualize something. So mechanism summary. Okay. Energy in visual spectrum focused on the retina has to be converted into electrical potentials or impulses by rods and cones or by chemical reactions. So impulses reach cerebral cortex through the optic nerve, and sensation of vision is produced in the cerebral cortex. Okay, and that is processing of visual sensation is explained on the basis of image formation, chemical and electrical formation. So image formation, chemical and electrical phenomena. So these are the three things that we are going to be looking at when we start looking at the visual process. Okay, later, that's the physiology. Now, the image forming mechanism. Okay, so process of focusing light onto the retina is supposed to be in a frequency of 397 to 723 nanometer. That's the wavelength that you're talking about. So not frequency, but the wavelength. So in order for us to be able to visualize something, it on the electromagnetic spectrum, it has to fall under 397 to 723 nanometers. So involving refraction or bending. So a property of the eye structure is can be seen there. So light rays are refracted by the biconvex lens, cornea, and fluids within the eye, creating an upside down image on the retina. So about all these areas being able to refract, refraction simply means bending. We already talked about it. So like I said, the convex lens is able to do that. The cornea and the fluids are also able to produce more refraction. Then let's talk about the refractory power. Refractory power is measured in diopter or D. So diopter is the reciprocal of focal length expressed in meters. So focal length of cornea is 24 millimeters and refractory power is about 42 D. Focal length of the lens is 44 millimeters and the refractory power is about 23 D. So now when you look on the far right, you're going to see that there's an image here talking about the visible spectrum. Okay, so this is where we have visible light. If you can see on the far right here, we have a visible light there. Yeah, if you see on this diagram, you're going to find that this is 352 somewhere, 750. This is where we can be able to see an image. If you remember, you guys, if you did watch The Predator was able to see images which were falling under this known as infrared, okay? Meaning it was even able to pick up heat energy. So the Predator used this, right? It was not using, whereas humans were able to see what we call the visible spectrum or the visible light. Yeah, I'm sure you have watched that movie before. All right. So now this diagram is just trying to show about accommodation and how a clear image can be seen. And if you can look at it at the end there, there's a stop sign. Then at the cornea, you're going to see an image that's upside down. It's up to the brain to interpret the image in a straight or upward position. So now refractive power is the greatest when the convexure of the lens is greatest. Okay. The human eye has a refractive power of approximately 60 diopters or D60D at rest. In the eye, light is actually refracted at the interior surface of the cornea and at the anterior and the posterior surface of the lens. The retina image is then inverted, okay? It's upside down. So now when you look at it, it's an image just trying to show. So like I said, there's light waves going there, there's cornea and there's a lens. So here it can be refracted. Also, there's a fluid there, it can be refracted further, also the lens and also in the vitreous humor. Okay, so this diagram is just trying to talk about the different areas where you can be able to perceive and how the waves of light or rays of light are able to be captured and how they can be able to be refracted depending in different types of areas of the eye. So if you can see here, it's saying there's air there, there's a cornea, aqueous humor, lens, and vitreous humor. So these are the different types of refractive index, how much it can be bent in order for us to achieve the perfect image on the fovea centralis. Okay, so this is optical axis, as you can see the straight line. Then the distance between the optical axis and the focal point is known as the focal length. Okay, and you have what we call the principal axis, as you can see. Then there's the optical center there. Then there is the principal axis. Then there's the prior rays of light, as you can see. So that's what this is talking about. So the distance between the lens and the principal focus is the principal focal distance. Okay, light rays from an object that strike a lens more than 6 meters away are considered to be parallel. Okay, more than 6 meters, they'll be moving as straight lines. The rays from an object closer than 6 meters are diverging and are therefore brought to a focus further back on the principal axis than the principal focus. Okay. Yeah. So now when you talk about accommodation, what are we basically talking about? I think next time when you meet, we can pick up the topic from here, then we can be able to do more. Okay, so please go through what we have learned and be able to come up. You need to understand what focal length is, focal distance, principal axis, and all those before we start what we call accommodation. Thank you very much and wish you the best.